Namaste. Hamisha Khush Vidya. I am JV Dr. Devi Trivedi, and today I am going to study before you a lecture on diseases of pigeonpea. Now, the first disease which we are going to study is wilt of pigeonpea, called as Fusarium udem. Now, these are the symptoms. Here you see the uh, field which is showing wilted plant, and we also see how there are red streaks, brown streaks on the vascular region of the root and stem. Now, the symptoms start from the early stage when the plant is 4 to 6 weeks old and up to the flowering and podding stage. The main symptom is gradual withering and drying of plants, yellowing of leaves and blackening of stems starts from collar to branches, and finally results in drooping and premature dying of leaves, stem and branches, and finally the death of plants. Now the vascular tissue exhibits brown discoloration, and on one side only sometimes there is partial wilting. The pathogen consists of hyaline septate mycelium. There are two types of conidia, microconidia and macroconidia. And the perinating structure is chlamydospore. These are thick-walled, terminal, intercalary body. Now the soil temperature favorable is 17 to 25 degrees centigrade, and the continuous cultivation of red gram, the same field, is also very favorable for this. The disease cycle consists the fungus survives in the infected stubbles, and the primary spread is through the soil-borne chlamydospores and infected seeds, and the secondary spread is through irrigation water and influenza. We use the trichoderma viridae for management of disease, and for this, the amount used is 4 grams per kg with a spore load of 10 to the power of 6 coliforming units per gram. Success: we should avoid mono-cultivation of red gram in the same field every year. Crop rotation with tobacco and mixed cropping with sorghum is very good. Resistant cultivars like Sharad, Jawar, Maruti, Pitta, Arar, etc. They are also very helpful. The second disease we are going to take is powdery mildew caused by Legionella torrens. Now, the main symptom is white powdery growth of the fungus on the lower surface of the leaf. So here you see the lower surface. And the corresponding upper surface shows pale yellow discoloration. Now, this white powdery surface is due to the conidophores and conidia. And when there is severe infection, this white powdery mass is also seen on the upper surface. And sometimes premature shedding of leaves also takes place, thereby devoiding the plants of all leaves. The pathogen is intracellular and absorption of nutrition takes place through haustoria. The conidiophores arise through stomata and they are hyaline, long, non-septate, thin, slender, and rarely black. Now they bear conidia matrices. Now these conidia are hyaline, single-celled, and elliptical or clavic. There is also black globose clostridia present with simple myeloid appendix. They contain 8 to 20 cylindrical asci, and each ascus contains 3 to 5 ascospores, which are hyaline and unicellular. Favorable condition for powdery mildew is always dry, humid weather followed by rainfall. The disease cycle is the pollination through Clostridia and ascospores, and they infect the lowermost leaves near the soil level. Secondary spread is through airborne pollution. The management is spray carbendazole 5 grams, 100 grams per hectare or vegetable sulfur 2 kg per hectare at the initiation of the disease, and 15 days later we repeat. Another disease we have is sterility mosaic disease, very important disease because it produces sterility in the plant. The causal organism is the pigeonpea sterility mosaic virus, and the typical symptom is chlorotic rings present on the leaves, complete sterility before flowering or complete sterility after flowering. So the plant gets a bushy and pale green appearance due to excessive vegetative growth. Stunting and prominent mosaic symptoms are seen on the leaf, and there is reduction in the leaf size. There is complete or partial cessation of flowering, leading to sterility. Now, three types of symptoms are seen. Either it is severe mosaic and sterility, or it is mild mosaic and partial sterility, or it can be chlorotic green spot without any noticeable symptoms. The pathogen, as already said, is Pigeonpea stereo mosaic virus. They are flexuous filamentous virus-like particles, and the weight is 32 kilodaltons. Size is 3 to 10 nanometer in diameter, and they are RNA species. Now, it is not transmitted by infected sap, it is transmitted by a mite, that is aerophilid mite, Escherichia pegina. Now, this mite contains the virus 12 to 13 hours in it, though the eggs do not transmit the virus. Now, the plants which are growing self-grown red gram plants, they are the perennial source of virus inoculation. Management practices include roguing of plants up to 40 days after sowing, spraying of monocrotophos to control the effects at 500 ml per hectare soon after the appearance of disease, and then repeat after 15 days. The resistant varieties, Pigeonpea or cultivars, can be used to manage the disease, or we have like ICP 6997, ICP 8861, Bhavni Sagar 1, etc. and many. So students, I hope you liked my lecture. This session was powered by digital version 2.0 of Jyoti Vidyapith University. Namaste and thank you very much. If you have any queries, please put in the comments box. I will be going to answer.